Good morning. Welcome to this second lecture of the Bioinformatics workshop, streamed to you from the snowy Finland. I hope that you enjoyed this workshop so far and that you have learned new and interesting skills. Before we start today's material, I have some technical notes. We have heard your feedback about the issues with accessing Moodle, and I was in contact with many of you, but if you need further assistance, please feel free to send us chat message here on this YouTube stream, or just mail me. I can reset your password or send detailed instructions how to log in if you need assistance. Just contact me in any ways, chat, email, whatever works. Also see Marty's note on the volume issue. We are on it, fixing it for the future lectures. Alright, with the technical things out of the way, let's start to look into the use of NCBI and Uniprot database tools, and I welcome again Dr. Martti Tolvanen.
Hello everyone, and welcome back to our Bioinformatics workshop. Today we'll continue on bioinformatics databases, and our main focus will be at NCBI on the east coast of the USA. The reason why we are focusing on NCBI a little bit more than EBI, actually we do not anything of EBI, but anyway, the reason is that NCBI BLAST is so much a better service than EBI's, so it's important to be able to retrieve sequences from NCBI, and the points I'm going to demonstrate at NCBI are just as good as the same things at EBI, but you'll see how the sequence retrieval works. Okay, so some databases are consorted to divide the workload and also to have some safety in case there's a volcano eruption or earthquake that takes down the DNA database of Japan. They still have 2 copies running at Hingston near Cambridge and in Bethesda, Maryland at GenBank. And the collaboration done between the global nucleotide sequence databases is called INSCD, and the way it works is that they share their data on a regular basis, so that no matter which one you use for your searches, you might be only missing something that was submitted during the last few hours. A similar collaboration exists between the PDB sites on 3 continents. The sites are slightly different even if the data is the same, so you might look around if you become a regular bioinformatician and see which site you like for particular services. And as I usually do, I drop regularly on both of the major sites, EBI and NCBI, but NCBI is where I go maybe 80% of the time. And if you're a scientist who submits data, so it doesn't matter which one you communicate with and just send your data, they'll take care that it's delivered between all the members. And just to revise from last time, the most important stable database resources that you should become familiar with are here, and you should be fluent with at least one of each class, a big DNA database, big protein database, protein structure database, and genome browser. And the ones in boldface in this slide are the ones we are going to be learning in this course. Now at NCBI, one of the nice features they have is that they have really integrated their data seamlessly so that a lot of the time you don't realize which service you are using or which database your data is coming from. You just click on a link, you get more information of the same thing, same gene, same protein, but you might be jumping between databases. And that's probably because they have a long experience in databases. Even at the time before the internet or any efficient internet, they were delivering the Medline database of biomedical literature, which came, I've been in a time when Medline was delivered on a huge pile of floppy disks, and then our librarian would be installing them at the library tabletop PC, and then we could get a fresh copy of Medline every few months instead of doing it in printed books that were several years out of date. But anyway, things have developed. Now we have the PubMed internet tool, but it is run by the National Library of Medicine, which is part of the same organization, actually a mother organization of NCBI. And so they've been really good with their sequence databases, and one especially admirable effort is the RefSeq collection. So when more and more data is coming into the databases, so you get more and more duplication of different research groups sequencing the same gene, etc. Having the gene sequence or protein sequence pulled from a full genome sequence or from a small scale independent entry. So these things give minor differences, and the curation work going into selecting the most representative and correct entries is saving a lot of time. So before RefSeq existed, I remember sitting down hours trying to figure out which of the 15 sequences is the good one for the protein I want to be studying. So that's a big plus. They do provide a large collection of full genomes, but I don't recommend NCBI as a genome browser. It's not as nice in the interface as Ensemble. But if you want to download genomes, so they are pretty efficient, and being in a different time zone is a plus for a European user, so that you might get your things done with less lag times than going to the EBI servers in the afternoon at European time at the same time when everybody else. And because these guys are the creators of PubMed and Medline, so it's no wonder that they are linking very nicely their sequence databases and the biomedical literature. You can do things like picking up articles which are linked to a sequence database entry easily in Uniprot and in NCBI, but it's even easier at NCBI. But the opposite doesn't work. So if you have a collection of articles which you have collected by whatever means, searches, combining searches, and then you can ask the question of give me all the sequences reported in these articles. So that's another plus. NCBI maintains the official taxonomy databases of the sequence world. They might not be the very best biological information of how species are related to each other, but it's a stable system which is avoiding the latest conflicts and latest changes in the hierarchy. They take them with a lag of a couple of years to see if they are actually accepted in the community. But anyway, it is a stable database which is fully documented, and it contains every organism that has any sequences in any of the DNA or protein databases. And this is the basis on which all the sequence databases are relying on, providing a controlled vocabulary as well. And then NCBI has some research going, and they've developed many innovative tools. As the most prominent one, the BLAST search engine is developed by NCBI guys back in 80s and 90s, and still running as the very best search engine of its kind. And another one which I like personally, because my focus is on protein, is their conserved domain database, which collects kind of protein signature or signatures of domains, smaller parts of protein, representing a compactly foldable separate entity that can be assembled together like Lego blocks having different domains. Should be a familiar concept to you, having different domains in a single protein. And a couple of years ago, they decided that CDD and domain signature databases in Europe, Interpro, PFAM, whatnot, have joined forces so that the Interpro signatures are now also part of CDD. But this is one of my favorite tools in trying to see if a novel protein sequence has any specific kinds of domains that have known functions. It's like trying to parse a sentence in a foreign language when you know that there's a verb and a noun which are the predicate and the object in the sentence. In the same way, I can say, okay, this protein has a carbonic anhydrase catalytic domain and a pentraxin domain which can bind to other pentraxins. So that's making sense on a higher level than trying to see that, oh, we have these 200 letters in the protein sequence, what kind of a protein function they might be carrying out. So these kinds of analysis tools and the analysis results are really important for us as regular users of the data to get the knowledge quicker instead of being drowned in the sea of amino acid and nucleotide letters. Now, we've been doing some keyword searches, which is easy. It's like Googling, and we've discussed the problem of these keywords not being always correctly typed. Google has pretty nice language technology that can correct the typos and do approximate matching, but unfortunately the sequence databases are just lost if you miss a single letter or if the author of the entry or the curator of the entry has made a mistake in typing in some scientific term. And the controlled vocabularies are a big help in that, but still you might need to look at your controlled vocabulary. For instance, you might look at the gene ontology molecular functions to figure out which are all the terms you want to use when you need to be looking at, let's say, innate immunity or signal transduction in innate immunity. But then there's another way of searching. Instead of depending on how well and how correctly the sequences are annotated, you can use the data itself, the sequence, and you can try to find sequences based on sequence similarity. And the thing we are going to be using a lot tomorrow is called BLAST, Basic Local Alignment Search Tool, and that's a lovely way of getting there, but that's for tomorrow. And no matter what is the annotation, if it says hypothetical protein and a string of 10 numbers and letters, so if it's similar to my query sequence, which would be let's say an alpha carbonic anhydrase, I can pick it up, and I can pick up this sequence and see that it has a 30% similarity, 30% identity with my query sequence. But then we might have problems if we have clearly a same sequence. Let's say we have human CA7, and I'm doing a search in vertebrate animals, and anything I pick up which is a true CA7 is most likely going to be annotated of CA7 because of the very high similarity. But anyway, when there's a gradient of similarity going from the best matches to worse ones, where is the cutoff? How do I know which of the sequences are true orthologs to my query sequence? I have a couple of tips for saving some time and saving work when you are doing bioinformatics research. One of them is that it's a good idea to register at the major sites, EBI, NCBI, Ensemble, Uniprot. Those user accounts allow you to save your search strategies and also to save some search sets. So go ahead and do that. The other thing is the need of documentation. Bioinformatics work, even if not dealing with tangible objects, requires to be documented to be publishable or usable in commercial research like pharmaceutical industry. And there are no fixed format lab notebooks for bioinformaticians, but there are many software solutions that might help you to document pipelines of your research, or you can just have a Word document to save crucial data, like when you access the database, what special parameters you had chosen for using some software, etc. And the old saying is, if you didn't document it, you didn't do it, and that holds for bioinformatics too. So if you don't see me doing it because for the sake of speed, it's not happening, but hey, I have the video to show exactly what I was doing here. But in your own case, now going back to NCBI, they have different collections which will be perhaps more relevant for the BLAST topic, but the default collections for doing anything in NCBI are what they call NR, which is supposed to be non-redundant, but it is not fully true. The NR databases are a union of different data sources they have available, and they are different for DNA and protein databases, obviously. And the only thing which is the non-redundancy is filtering that 100% identical sequences of the same length are eliminated. And in case of proteins, the NR database would be a union of RefSeq and GenPept, Uniprot, and PDB sequence entries combined, with priorities pretty much in this order, so that the best quality RefSeq entry is selected out of multiple identical ones. GenPept entry, because it's one of their own making, is chosen over a Uniprot version, and Uniprot, if it happens to represent a different sequence, comes as a small percentage of additions. And PDB very rarely has anything that is not already included in the actual sequence databases. But anyway, the redundancy is very real because of minor variants. For human proteins, you have different allele variants, maybe sequencing errors coming from different projects, and that means that for any protein you easily get 5 or 6 or even dozens of entries representing supposedly the same sequence. And with RefSeq, you save a lot of work by just getting a single sequence. The NR entries, when coming from some of the outside databases, they retain their accession numbers, accession codes. So for instance, the Uniprot code is there, but they add a GI number from NCBI in order to have a unique identifier for everything, to be sure to have a unique identifier. And in cases where the sequences are identical, so they drop the Uniprot version, and it's important to know that if you want to filter your data for identities, so it's not enough that all sequences have different accession numbers. If they come from different databases like NCBI and Uniprot, the same sequence might come up as two entries which have a different ID and also different annotations because of a different history. Now, I mentioned the idea of doing your advanced search afterwards, and the tool I can recommend for that is the data filters they provide on the left-hand side of the result page, as shown here. So I made a quick and dirty search with the word zebrafish carbonic to find carbonic anhydrases in zebrafish, and as a result, I can see that my result set contains 19 bacterial sequences, which are certainly not what I wanted. So just clicking on animals, I could remove those bacterial sequences. And if the list contains partial sequences, I might get better quality results by choosing RefSeq. Zebrafish is one of the model organisms, so I could expect this to be pretty good. Or else I could use the sequence length filter here to give a minimum length, the length I know I'm expecting for carbonic anhydrases, which would be more than 240 amino acids. Now, regarding RefSeq, we've talked about them in a few occasions. These are sequences as said to represent the best possible version of each transcript and each protein, and even sometimes genes, but the gene project is still unfortunately not very long gone. But still there will be splicing variants as alternative products for the same gene. And now it's good to look at the accession numbers of your data. You can immediately spot RefSeq entries by them having NP underscore and numbers, maybe 6 or 7 numbers, or XP underscore NM underscore XM underscore. Now, P is for proteins, M is for mRNA, so they are transcripts. And then this N means known entries or reviewed entries, whereas X is sequences which have been derived from automatic genome annotation without any checks for transcript support or correctness of the prediction, no touch of the human hand. So basically less reliable, but for many species, that is the only thing you can get. Now in NCBI search tools, we looked at the data filters on the left. There's another thing which is on the right-hand side of the web pages. And I brought up this snippet of the page because it's not right there at the top. You need to scroll down the page a few screens to find this section of related information, and this is really valuable because you can access other things which are related. This comes up when you are looking at a single protein entry. So then they will have links to nucleotide and PubMed. If you are looking at a nucleotide entry, there would be protein, PubMed, taxonomy, etc. So you have fuller access to cross links to other NCBI databases and sometimes even other databases. Okay. As to saved searches, I've found that the My NCBI service is the one that I've been using most for saving searches. It might be just because of having found it first, so that's where I like to save my searches if I have something needing. This is the end of the theory part for today, and we'll move on to the hands-on demos. This is a quick list of things we'll do at NCBI, and you might refer to this slide from the PDF of lecture notes, and likewise for Uniprot, these are the tasks we are going to do. So let's move on and do them. All right, let's do a quick demo on a search on carbonic anhydrase and one of its synonyms, carbonic dehydratase. We can see that we get about 500,000 hits in NCBI protein with the first term. And we can see that carbonate dehydratase is a common term because it's a top-level term for the protein family in bacteria. And for the carbonate dehydratase, still we only get about 70,000 terms. For the other one, now we want to see if the second result set is totally overlapping or contained with the first one, or whether they are totally separate. And we can do that pretty easily with the Boolean combinations of these result sets. And we find this in advanced searches, in the advanced search. So we can see that we have the searches made previously as number 5 and number 6. In order to combine our research sets, we can use the number symbol and the actual number as a search term. And if I click edit here for building search, I can add our first searches, number 5, and then to make a union of the two sets, any proteins mentioning either carbonic anhydrase or carbonate dehydratase. this would perform the search. Clicking the search button, we get there. But I've already done that, and what we have here is the results, which show that if we combine both terms, we get 560,000 instead of just the 518,000 with carbonic anhydrase. And just 27,000 entries mention both of these terms. So doing this combination of the two synonyms seems to look good. And going to the result set of the union, 562,000 terms. And if we limit on refseq, clicking to refseq here on the right, now we are down to 123,000 sequences. Well, I'm not going to do a multiple sequence alignment. For one thing, these would be divided into multiple protein families of alpha, beta, etc. carbonic anhydrases, but we can see here in the list of taxonomic categories that vast majority of these are bacterial, because bacterial genomes have been sequenced so frequently, and each one has at least one of them. Many of them have several. So with hundreds of thousands of bacterial genomes, it's adding up. And even in animals, we have 24,000 sequences, so that would be quite a lot to study and classify if we wish to do that. Now let's do another demo. Now that we have combined the sets of carbonic anhydrases given in two different terms, now let's get the best possible search to see how many protein entries we can find from the Australian ghost shark or elephant shark. And for that, we would choose organism from the list of fields, primary organism, and type the Latin name of this cartilaginous fish, and we want to add to history the number of entries found with this. So that's about 52,000 proteins.